السلام عليكم ورحمة الله تعالى وبركاته. In the series of lecture concerning the course Applied Pharmacognosy, which is designed for fifth-year pharmacy student, in this line lecture, we will talk about collection and extraction of marine organisms. Marine environment contain over 80% of the world plant and animal species. And in fact, 50% of naturally occurring compounds obtained from marine organisms reported in literature are biologically active. On the other hand, marine pharmacognosy is considered as branch of pharmacognosy which is concerned with natural compounds that having medicinal value and obtained from marine organisms. The extraction and collection of marine organisms can be carried out using various methods depending on the nature and type of organism and compound to be extracted. Because marine organisms and marine compounds have special features. Most of marine natural products occur in marine environment are present in low quantity. Also, these marine natural products characterized by instability and are highly polar. Because of these properties, special extraction and collection techniques is required. In this lecture, we will talk about some important example of marine organisms that use for extraction of marine natural products. And secondly, in this lecture, we talk about collection and storage of marine natural products and marine organisms. And at the last or at the end, we talk about extraction methods used for extracting marine natural products from their natural sources. As a general introduction for the topic, and based on the published data on marine organism and marine pharmacology, they found that marine flora and fauna play a significant role as source of new nuclear entities. And also, in the literature, the ocean of the world contain over 5 million species in about 30 phyla of organisms. And marine environment also contain over 80% of the world plant and animal species. That means the marine environment contain a higher and various type of plant and animal species compared to the land. Unfortunately, half of marine natural products that are discovered and reported in literature are biologically active. That means, based on this information, the marine organism having a potential, considered potential source of biologically active compound that require further studies and research. Marine pharmacology is defined as a sub-branch of pharmacology that deal with or concern with naturally occurring compounds obtained from marine organisms and this compound having medicinal or therapeutic potential. And generally, the medicinally active compounds obtained from marine organisms such as bacteria, fungi, algae, sponges, worms, and so on. One of the important online databases that concern with marine organism and marine natural products is known as marine-led database. Fortunately, this database contained more than 4,796 genera and species of marine organisms, which is considered a source of novel marine natural products or NMBs. An example for marine organisms is considered as a source of NMBs, including uh, bryozoa, which belong to considered as حجازيات, nudibranchi or عاريات الخيشوم, مولوسكا أو الرخويات وتونيكات نوع من القلاليات soft corals نوع من أو ما يعرف بالشعب المرجانية سبونجي سفنجيات والكون سنيل اللي هو القواقع المخروطية أو القوقع المخروطي وبيسايد الورمز أو الديدان. Also, green and brown and dead algae are considered as one of the important marine organisms that having a number of natural occurring compounds. Example of green algae that having therapeutic importance: Codium green algae, Caulerba genus, Enteromorpha genus, and Olva genus. For collection and storage of marine organisms and marine natural products, the important information is that marine organisms require special handling techniques because they are uh, contain higher moisture content, which can be decomposed, and that concept also can be decomposed during collection and storage. Another important thing for collection is the wearing of or the use of protective equipment such as gloves and eye protective because marine compounds are extremely toxic. For example, many organisms such as sponges or spongiat are highly irritating or contain highly irritating compounds that cause immediate itching. So the precaution for collection and storage of marine organisms is based on the fact that marine organism and marine natural products are unstable, which can be decomposed easily when exposed to air or high temperature during collection and storage. And on the other fact, and the another fact is that these compounds are highly toxic and highly irritated. So protective measures should be considered during collection and storage of these marine compounds. As we deal for collection of plant sample, their important information should be recorded, and herbarium sheet should be uh, prepared, and partial shipment should be uh, prepared also. For marine organism, also we follow the same uh, techniques and procedure for collection of marine organisms. And the information on organism and place of collection should be carefully recorded to facilitate recollection of the sample and the subsequent taxonomic identification process. The presence of associated organisms also should be noted and reported because it is very common to find worms, mollusks, and even small fish living with larger marine invertebrates. All this information should be recorded and noted for identification purpose and for recollection purpose also. Here an example for collection sheet or collection record sheet should be used during collection. It is necessary to have a number for each collection, write collection number, and decide if it is a new sample or recollected or old collection. Also, the date of collection is important for identification and for recollection to determine the season of collection. Also, the common name of organism collected, also the name of collector. Also, it is very important to take a photo of the collected sample. Also, or in fact, two photos underwater and when removed from water or above the water. Also, site of collection should be reported, and the important information for site of collection is that the depth in meters, where the place where you find the organism, also habitat. Is it in the ocean or in sea or the river and so on, or even in the abundance also should be reported, and here abundant, very common, containing high content, or common, or occasional, or rare. So the abundance should be uh, predicted or indicated. Also, weight in kilograms should be reported. For appearance of marine organism, we should write the color or the colors inside or outside of the organism, or also the color in alcohol, because the color of some marine organism can be changed during collection. Some organisms having color when they are in water and color when they are outside, or we change the salt concentration of the water within which the organism occurs. Also, the shape of organism should be recorded. Have is it interesting, tree-like, mushroom-shaped, uh, fluid, serum-like, and so on. There are some of many terminologies can be applied to describe the shape of a marine organism. The texture also is very important, as in plant part. Is it slimy, fleshy, waxy, shiny, and so on. Also, the surface should be described. Is it smooth or spinned or lumpy, fizzy, and so on. Exudate, if it occurs, some exudate should be noted. Is mucus or slime or mud or stones? Is it sticky or is it colorless or colored? Easy details you have to decide هل هي ممكن can be fractured easily or cut it easily, yes or no. And further notes should be also reported if any crystal formation is not recorded in sheet, and your note in your collected organism should be recorded. For the voucher specimens, which is important for taxonomic process, and we mean by voucher specimen is العينات المرجعية اللي هي مهمة لتصنيف العينات. So the voucher specimen should be prepared by taking a small section from the tissue of marine organism, and usually maybe in the range from 2 to 5 centimeter in length. And also it's important to preserve the voucher sample in solution of 10% formalin in seawater. For algae specimen, if it is green or brown, and algae should be collected and preserved in 5% solution of formalin in seawater. One of the important measures or things for voucher specimen, beside the preservation methods, uh, the specimen should be representative of entire organism and include as much tissue relevant to taxonomy as possible. For example, for sponges, the spongiat, we use both exo and endosome, which are essential for accurate identification. And for tourniquets اللي هي الغلاليات والsoft porous المرجان, often we take the whole organism if it's not too large, and also important to take the root of this tourniquet and corals. So after the specimen reaches the lab, we should remove formalin solution, can be replaced by addition of 70% ethanol for long-term storage. Now for collecting marine organism for extraction purpose, it's very important to note that the amount of organism to be collected should be relevant to their abundance. And the ideal size for sample is in the range of 1 to 2 kilo اللي هي in wet weight. Usually equal 100 to 200 gram dry weight, because the moisture is present in higher level in marine organism. Also, the complete harvesting of organism should be avoided, and if only a single large organism is available, a part of it may be collected. ضروري جدا ما نجمع كل المارين اورجانيزم موجود في المكان أو في المنطقة المحددة، لكن إذا كان عندنا وجدنا single organism ممكن ناخد جزء منه فقط، هو كافي for studies. والهدف من كده إنه نحمي البيئة البحرية خصوصا للأنواع النادرة أو المنقرضة. After collection of acquired marine organism, this molecule should have to be stored and transported to the lab. And actually, marine collection take place on remote places, which is very far from laboratory facilities, and the laboratory facilities in this place may be few or limited. Therefore, the collected organism should be dried or extracted or frozen immediately to reduce spoilage and chemical degradation. Uh, some type of marine organism require special handling and special attention. For example, some type of sponges known as Virongida sponges. This type of sponge begin to degrade and polymerize immediately upon being touched. When it is touched, change it in size, polymerize, and become degraded as defense techniques or defense mechanism. This indicates by rapid color change from white to yellow or orange to dark blue, even when maintained in seawater. This is important information should be noted when collecting uh, marine organism and when you tend to store these marine species. So, in order to avoid the degradation of marine organism and marine compounds, after collection of these species, the sample should be immediately freeze dried or lyophilized to prevent any chemical degradation. If immediate lyophilization is impossible because the lack of instrument or lack of facility, laboratory facilities, the sample should be kept on low temperature, usually from minus 20 to 0 Celsius until freeze dried. And after freeze drying, the sample stored either at room temperature or in freezer. Alternatively, if it's impossible to freeze dry, freeze drying or storing the collected sample in low temperature, sample can be immersed directly in a mixture of water and ethanol solution, usually 50 with variable volume percentage for 24 hours after which the liquid is discarded. So this is considered as transitional storage measure to transfer the sample from site collection to the lab facilities. After addition of or after immersion of sample in methanol and water preservative, the dump organism are then placed in high density polyethylene bottle. For example, there is Nalgene bottle having wide mouth, considered as the best solution for storing of marine organisms. And this bottle aid to transport the organism to uh, to the lab in temperature about 27 degrees, 27 Celsius, or considered as the ambient temperature. Actually, the sample preserved by using ethanol and water and place it in a specialized bottle for transport. It may remain good for 2 weeks in tropical conditions with no significant loss of secondary metabolites or secondary content. So the addition of methanol should occur immediately after sample reach the lab to reduce the degradation process. This is for dam organism. For algae and some organism known as Gorgonians or Gismin المرجانيات are often air dried and stored at room temperature. This type of organism did not require uh, freezing or addition of preservative. Can be dried and stored at room temperature without any change, or can be preserved in good condition by using this dry technique. After successful collection and storage of the desired marine organism, the next step is extraction of these samples. Actually, there are three extraction strategies used for isolation of marine natural products. And selection of extraction strategy depend on three main criteria. One of them is the aim of isolation process, and the second is availability of the required facilities and the intrinsic advantage and disadvantage of the procedures. The three extraction techniques for NMBs include maceration, NCI method for National Cancer of Institute, American Institute of Cancer method, and the third one is using SFC, super critical fluid chromatography. For maceration, it's similar to maceration process used for natural products from plant, and it involves several steps. One of these steps is grinding of sample if it is dry sample, or cutting into small pieces if it is damp or wet organism. After grinding, the second step is maceration of sample with solvent, followed by filtration or centrifugation to separate the extract from the debris of tissues of the organism. Stirring or sonication also can be applied to increase the diffusion rate and to accelerate the extraction process. Usually for maceration, to extract marine natural products, we use methanol or ethanol as solvent of choice, because almost all marine natural products are water soluble or polar compound. The second extraction method used for marine organism is the NCI method, or National Cancer Institute method. It's worth mentioning that the NCI Institute of Cancer uh, have extraction or screening program for natural anti-cancer from marine organism, and continue for several years, and end with a number of very active anti-cancer agents from marine and plant sources. In this method, the frozen sample, a freeze-dried sample, are ground with dry ice. Dry ice is carbon dioxide, and then extracted with water at 4 Celsius temperature. Secondly, after grinding the sample with dry ice, the aqueous extract is removed by either centrifugation or lyophilization. The mark left after centrifugation and lyophilization can be successively extracted with methanol and dichloromethane. 1 to 1 ratio, followed by methanol 100%. And this step is to ensure all the active constituents are extracted from the mark. Then the organic extracts are combined and concentrated under vacuum. That means the fraction obtained by uh, aqueous water and dry ice, and other fraction obtained by methanol dichloromethane solution, and fraction number 3 obtained by methanol extraction 100% are combined together. The NCI method of advantage over maceration is that the use of lyophilization of aqueous extract reduce the risk of heat degradation that may present during extraction process. The third extraction method used in the field of marine organism extraction is SFC method or supercritical fluid protocol. In this method, carbon dioxide is used as solvent, and the advantage of this extraction the yield or the extract obtained after removal of carbon dioxide will become uh, dry and free of solvent residue that may occur with NCI method and maceration method. The another advantage of SFC protocol is uh, the low viscosity of carbon dioxide as solvent, and it is become a gas after removal of overall pressure and temperature. Also, the other advantage of SFC is superior mass transfer properties, means carbon dioxide is inert, and the solvent, the extract is solvent free of after removal carbon dioxide. Also, carbon dioxide have good sorption power and ability to penetrate microporous material and offer fast and effective way for extraction and subsequent solvent removal. But the disadvantage of SFC protocol is that the equipment is sophisticated and it requires experiment, so required experience to be operated and to choose the best organic modifier. Sometimes we need to add organic solvent such as methanol or ethanol to carbon dioxide as solvent to increase the polarity and aid the separation of the active constituent from grounded tissues. That means the use of organic modifier. So, organic modifier means addition of uh, polar solvent or organic solvent to carbon dioxide to increase the polarity of the solvent and to accelerate the extraction process. The extraction of macroalgae now is given as example for extraction of marine organism. And as we mentioned previously, macroalgae can be dried either at room temperature or, or by freeze dry, and some can be stored at room temperature without degradation of its constituent. In this example, macroalgae can be extracted by using Soxhlet extractor, which considered as a modified form of maceration, which involves maceration and continuous flow of solvent or medicinal. So the first step for extraction of macroalgae is drying of sample, either by localization or room temperature or air-dried. And the second step, the dried sample can be grinded either by using mortar and pestle or using Wiley mill. And Wiley mill is a type of mill that occur in soil laboratory used for cutting the plant sample or the soil material. After grinding, the sample is sealed by using sieve, mainly 2 mm sieve. And the third step after sieving, the grinded material can be extracted successfully by using solvent of increasing polarity. Uh, for example, using of hexane as less polar, followed by extraction of dichlorohexane, and then followed by extraction or by using methanol. And extraction process take place in Soxhlet extractor. Sample placed in Soxhlet, connected and placed on the heating source, and then the flow of mobile phase or the solvent is started. The alternative method for extraction of macroalgae can be carried out by using maceration. We follow the same steps. Dried or frozen sample can be extracted by maceration using a high-speed blender. Then the extract filtered by using Buchner funnel under vacuum to reduce the degradation process of active ingredient when can be filtrated without vacuum or under high pressure. After filtration, the mark or the algae tissue can be returned to blender and extracted again to ensure the removal of all active constituents from algae tissues. In fact, both Soxhlet method and extraction by maceration in blender, the solvent is continued until no further color is extracted, which is known as exhaustive extraction. Continue extraction until all active ingredient are extracted or removed. Lastly, after extraction, the extract concentrated by distillation and the reduced pressure. At the end of this presentation, I summarize the important point. Uh, One of them is that the MMBs are considered as bioactive compound isolated from marine organism, and marine organism present in oceans or in marine environment can constitute about 80% of plant and animals that occur in the land. And isolation of extraction or collection of marine organisms require special techniques and methods depending on the nature of organism and active constituent to be extracted. Because the fact that marine compounds and marine organisms are characterized by instability and the presence of low amount of active ingredient, and then dealing with organisms that live in environment with high salt and water content.